Run.
The Bostonians who started Grey Thumb always had their meetings with places with lots of good beer, so they probably had a better insight than I do. But this is Grey Thumb, and it stands for sort of green thumb is a gardener of Terran biology. Grey Thumb sort of gardener of technology. You know, the products of they came up with this several years ago, and they formed it as a homebrew computer club for artificial life experimenters, practitioners. So if you know what the Homebrew Computer Club was, it was this group of geeks that met in the garage, and then they met at Stanford starting in 1975, and they brought in their Altair kit computers and they tore them apart and they showed us stuff off, and a couple of the kids were Steve Jobs and Steve Wozniak that were showing off what they built, and so it was a sharing kind of forum, and out of the Homebrew Club came the whole computer industry. Chunk of it came out of it, microcomputers. And so I don't know if huge industries are going to come out of this, but certainly a lot more creativity can be had if you have a social group to meet with, especially around a topic as strange as this one. My interest and background in this is I've built some kind of artificial life-like systems in the mid-'80s when I was a graduate student at USC. There wasn't a field of artificial life, so they were called cellular automata, and I built them on VAX computers connected to the ARPANET, or the internet at the time. And then in 1996, I formed an organization called Biota, which is a subset of another organization I formed called the Contact Consortium, which does all this stuff to do with avatars and virtual worlds. And I always felt that inside virtual worlds might be in a chance for a rich enough little ecosystem for artificial life experiments to happen. So that's why biota.org was formed. And we had 4 conferences starting at the Burgess Shale in 1997 and Cambridge, England. It was quite a great conference series. Really interesting people like Richard Dawkins involved in it. And then Tom Barbalet, about 2-3 years ago, started our podcast. And the podcast is sort of the modern way to do a conference, is to bring in thousands more people. And we podcast initially all our old conference presentations, and then we went to interviews, and now we have a live radio show once a week that you can call into on the regular phone, and it's recorded, and it's called Blog Talk Radio. And it's been a lively, amazing run of great conversations of historic figures like Steve Grant who come back into the fold, to new projects, new people, new visions, paleontologists calling in, immunologists, people from different fields that have a passion for this idea. So that's where Biota is. And so back in March, Tom and I thought it was time to go and talk to Grey Thumb. So I went out and did a meet and greet and had lifted some beers with them on March 3rd, and then we established a really good friendship partnership with the Grey Thumb, many of whom had been listening to the podcast. And I suggested we start to start chapters. So I said, well, let's start a Silicon Valley chapter. They started a London chapter, and now there's a chapter in, I think, starting in Brighton. To this day, June 24th, is establishing a first chapter meeting in Holland. So it's spreading. Whatever it is, it's spreading. And one of my personal interests in all this is the PhD proposal, the work that I started at USC in 1985. I always wanted to get back to it. And I was offered a position at a university in London as an external graduate student to do a PhD, or a practice-based PhD, about a year ago. And then I basically decided to go for it back in January, wrote up the proposal, and it's actually a proposal to continue the work I started in 1985. And my old professor from USC wrote a letter to the university so that I could be classified as a continuing PhD student, which is sort of funny with a 22-year interval. It's a large, but I showed that there was a body of work in between, and mainly was that processors improved and it was possible to do the kind of thing that I was thinking of. And that work particularly has morphed into an idea that we as artificial life practitioners can't have a healthy medium if we build things in isolation. If we put all your energy to build a little walled garden experimental universe, you'll run out of energy or resources or daytime hours, nighttime hours to continue it after a while. If these systems were connected into a larger grid, where even though they're different, they have different properties, objects can be sent back and forth between the environments. And objects might be creatures or just stuff that is flowing, stuff with APIs and affordances that appears goes from one world to the other in kind of an encapsulation, and then instantiates in the new world and does what it tries to do. That will unite our efforts to create, to create evolutionary systems in ways we can't even predict. You'll have that fantastic, weird side effect of look at all the weird stuff that's happening that we could never predict. So I came up with a name for this vision, which is the Evo grid or evolution grid. Why grid? Well, evolution's obvious, but grid computing is sort of the basis now for a huge amount of what's done in science and in multiplayer gaming. So, Second Life is basically a huge grid of machines with these portal viewers called the Second Life browser that's looking at different segments of the grid. World of Warcraft's the same way, and then there's gene grids, and SETI at Home was one of the original grid systems. So if we had an artificial life grid or an evolution grid, we might be able to really achieve something as a group, and all our A-Life boats would rise along with this grid. So that's actually was the proposal for the PhD work, and they accepted it, maybe because they didn't understand it. So I had no risk of them rejecting it. And I'm going to recruit a great community of people. One of them may be Richard Dawkins, because he spoke at our second conference, and I went to have tea with him. I can't have tea with him anymore, probably, but in Oxford about 5 years ago, and described this vision, and he really liked it and wanted to be part of it. So I'm going to go try to have tea with him again. You can't get on his calendar anymore, but and see if he would want to be an advisor to the project or somehow associated. And we'll get all the original names of people that have, Carl Sims and all the original folks, Tom Ray, like that, could be part of it too. And so there's one more little piece of the puzzle that's come in, which is talking with people like Richard Gordon. Richard Gordon is an immunologist in Manitoba in Canada. He was at Digital Burgess, which was our first conference where we climbed to the Burgess Shale up in the Canadian Rockies. And he's an amazing guy. And he and another professor are putting a book together, which is going to be published in October by World Scientific in Singapore. And it's about 25 contributing authors on questions of God versus science, God, creationism, intelligent design, evolution. And it's got chapters written by philosophers, by a cardinal, rabbis, evangelical thinkers, biologists, etc. The head of the Vatican Observatory has a chapter in there. So it's going to be quite a book. It's 900 pages, so it's not the kind of bedtime reading, but it's going to be coming out. And he's whenever I present the Evo Grid idea to him, and this is a concept for you guys to really think about. He said, how do you know that you have real evolution going on if you have all this artifice, if you have created a structure of genes already? You said, well, let's create a body and create a phenotype-genotype relationship and have a creature made. That's kind of, you're already sort of halfway there. You're already putting a lot of invention into it. Is it working? Oh, there's still...
Not yet.
Okay. I'll keep talking. Maybe it'll work off that machine. So, just to finish the idea, he was challenging the assumption. He's saying, really, when it comes down to it, and his book chapter is called the artificial origin of life. He's talking about artificial life, saying the only way you can be kind of have the litmus test, pass the litmus test of whether you have a system that is exhibiting real properties of biology, is if you start from scratch. If you start from, there's Fred Hoyle wrote something in the 70s or 80s that talked about having a tornado go through a junkyard of parts and suddenly the 747's assembled. Well, sort of taking off on that challenge, starting with the origin of life problem, a jumble of chemicals, and if you run a chemical soup long enough, you get vacuoles form with bilipid layers, you get stuff inside that can repair the bilipid layers, and you get a blueprint that can be used to copy the vacuoles, et cetera, et cetera. Sort of, this is something thought about in the origin of life community all the time. But what if you created an arbitrary set of statistical simulations and ran them with large grid or multi-core processors, and could show that replicators would emerge within that and persist and develop the properties we might call lifelike? Richard thinks that is a much more grander challenge that our field could take on. It's that really low level from low. What does it tell you? It tells you that yes, in principle, this could happen in a simulation, and if you could create enough, is it hard or easy? It casts some light on the origins of life a little bit, but it will teach us a lot about how hard it is, about how we might look at it in chemistry, because we may not be trying to simulate the chemical world because we can't. It's too much computation to do that. This is just ridiculously computationally intensive to simulate normal chemical interactions. But it might be that that's kind of a good crisp thing that we could get our teeth around as a community, is to do that origin of artificial life, stepping really back really far. Now, of course, what are the starting parameters? So if you're an artificial god, and you're playing, we may have found a solution.
We're ready to go.
Turning 90 degrees, we find a solution. So the last thing is, so you'd say, well, you're just an artificial god, you're just setting the starting parameters for this soup, and of course things come in. And it's at the center of debates over religion and creation and origins and things like that. It's just such an interesting, it's an open-ended, interesting project that one could take on. And it may be that it's a big science project. It's maybe not a hobbyist project, maybe it's thousands of multi-node computers and some wacky person who tries to get funding for this and keep it going, and then one day, or do you need to recruit 100 million home computers, and the one that generates a replicator, you could win some prize. It's like SETI at home, first person to find a signal. Anyway, but that, I'm rambling on here, but that's the idea. Is it's something to talk about later, but do you go for this really fundamental grand challenge, or do you stick with building things that are lifelike and see if evolutionary properties appear in them and maybe try to grid them together? So, and I'm heading to London in 2 weeks from Saturday, a week from Saturday, to start this whole crazy enterprise. And that's my report. Take it away, John. John Cumbers, Brown University. Prior to that, in the UK, presumably.
Right.
Somewhere.
London.
And then last 3 months at NASA Ames.
I've been at Ames for a month, and I'll be here for 3 years or more, doing research on synthetic biology and how it will apply to space settlement, biological ISRU, and ISRU is in situ resource utilization. So in situ, where you are, resource utilization, utilizing the resources for manned missions in space. And in space, we mean an asteroid, primarily the Mars or the Moon at the moment. 5 years ago, I was a software engineer. 3 years ago, I was a bioinformatician. Now I'm a biological engineer. So I know where you're coming from if you come from the software world. And I'm hoping to keep everyone with me when I talk about what we are doing with biological systems and how we're hoping that we can treat them the way that we've been treating electrical systems and computer systems, in that we are now at a situation where you can fairly easily program a computer to do whatever you want it to do. We're only just at the beginning stages of programming biological systems so that you can do what you want them to do. The talk is interwoven with cool pictures of space, inspiring you of what we're going to be able to do there, with some fairly simple biological systems that we've managed to control. And I'll show you some of those examples. So this is the grand vision, that whenever we travel to another place in the solar system, at the moment the mentality is that you take everything that you need with you for the trip. And that's got to change. That mentality's got to change to one where you don't take it, but you make it. And why is that going to be a possibility? We know it's going to be a possibility because if you look all around us on Earth, most of the building materials, maybe not most of them because stone isn't, but most of the wooden building materials, most of the things that we see around us, human beings, everything is made out of carbon. It's made out of carbon dioxide that used to be in the air. The carbon is now fixed into things that we can see around us. So there's no reason why that couldn't happen wherever we go, that there's a carbon source. The carbon dioxide content of the Earth is negligible, it's very small, less than 1%. On Mars, the carbon dioxide content is 97%. So there's a big problem at the moment in that we're putting too much carbon dioxide into the air from burning fossil fuels here. If we went to Mars, there's a huge amount of carbon dioxide in the air, and I'm really happy about that because I'm going to build things from that carbon dioxide that's there. So what you can see is a schematic of a photobioreactor, which is a bioreactor, which just means a place where cells can grow, and a photobioreactor meaning that the input to that system is light, and the output is whatever you want it to be.
Sounds like an aquarium.
Yeah,
Maybe
I guess, yeah.
John, if you're not going to mention the fact that the Earth's atmosphere is a bioformed thing from algal mats and stromatolites.
Sure. So the current theories on where what the Earth's environment was like before and what it's like now, which is it was before lots of carbon dioxide like the Martian atmosphere, and it is now 23% oxygen, 70-odd% nitrogen. And the reason being that there's a lot of carbon dioxide, the current theory is because of all the explosions during the formation of the Earth and during the formation of planets that throws out a huge amount of carbon dioxide. And it's small, single-celled, photosynthetic organisms that have taken the water, split it, and released the oxygen into the environment, which is now one multicellular life like us couldn't survive because we need that oxygen in the environment. There's work in NASA going on about that it may not be single-celled algae that did it. It could be a lot to do with compression of different rocks that produce the release of oxygen, which I was just learning about recently and didn't know about. So I'll speed up because I wasn't planning to talk so much and whiz through the slides. And please interrupt as we go, particularly if I've said something that I've taken for granted and isn't granted, because I don't know this audience. So I like to set out a goal. I think Kennedy did this very well at the beginning of the 60s. He was talking about sending a man